day everyone. This video lesson on quarter 3 week 4 will teach you about mutations in the DNA. This is Namjoon. Ready your notebooks and pen as we explore together mutations in the DNA. To start, let's have a fast drill for 15 seconds. As fast as you can, try copying the letters below. Did you get it perfectly? Well done! Well, some of you has copied the letters perfectly on their notebooks. Some might also have committed errors. It's fine. In fact, even the most complicated and programmed body process also does this. Recall from our previous lessons that the DNA undergo processes such as replication, transcription, and translation to be able to express the traits they are intended to through the process of protein synthesis. However, during these processes, mistakes or alterations can also be encountered. Consider the illustration above. Say, for example, in replication of the DNA, nucleotides are complemented mistakenly in a point on the process. These changes can affect a pair of nucleotides in the DNA strand or even a smaller segment of the chromosome as a whole. This further results to changes in the messages or traits that our DNA will produce. What causes these mutations? Mutations can occur naturally. This can occur upon inheriting certain mutated genes which are present from the parent's genes. Some come through chances which are of course rare and uncontrollable. However, there are substances that can induce these mutations. They are called mutagens. Common mutagens are in harmful chemicals such as carcinogens or cancer-causing substances, or even from the smoke of your cigarette. Radiation, such as ultraviolet and X-ray, also cause these DNA alterations. Lastly, some infectious agents, such as specific strains of bacteria and viruses, are just few of the biological mutagens possible. How does mutation takes place? We can consider here three types under a two broad category, namely point mutations and base pair alterations. Point mutations include substitution, while base pair alterations can be through insertion or deletion. Substitution or point mutation can occur when another nucleotide is placed for another nucleotide. Let us consider this illustration. Given a DNA sequence, say a guanine is replaced by another nucleotide, adenine. This results to a change also in the complementary base from cytosine to thymine. Insertion, on the other hand, occurs when a nucleotide is added within the sequence, thus changing the whole template for protein synthesis. The addition of a base shapes the template rightward, causing a difference in the reading that would occur until protein is synthesized, since RNA bases are translated by threads. This changes the reading frame. Say from adenine and thymine base pair, another thymine is added before adenine. This would correspond to an adenine as complementary base. Observe how the DNA sequence shifts to the right. When a nucleotide is removed from the sequence shifts to the left, varying as well the length of the DNA sequence. This is called deletion. When the base pair adenine and thymine is removed, observe the shift to the left and the reduction in the length of the sequence. How do these mutations affect protein synthesis and the traits expressed? Let us watch this simulation. This is a normal DNA strand to undergo transcription and translation. Take a closer look to the protein synthesized. What if on the same DNA changes occur? Remember some possible causes of mutations. When the adenine thymine base pair is removed, translation and transcription continues accordingly. See now how the protein made looks like. Will they still look the same? Good observations. From the simulation above, we can see how the changes in the DNA affect proceeding processes until the synthesis of protein molecule. These alterations in the DNA changes in the base pairing of the nucleotide. It also changes the resulting RNA from the transcription. More so, changes in the amino acid sequence number and length can be observed. Lastly, changes in the traits expressed in an organism will be affected. A change in the DNA sequence might not also affect the protein to be produced, since some amino acid sequence can be derived from multiple codons. This is called silent mutation. Consider the sequence guanine, cytosine, and cytosine. It will be translated into amino acid alanine. However, when we change or when we alter the last nucleotide base, according to the chart, we will still have alanine as its amino acid. Now consider this sequence, cytosine, guanine, cytosine. This would be translated into amino acid arginine. However, when the cytosine is replaced with guanine, it will now translate into glycine. A nucleotide is altered producing a different amino acid sequence. This is called missense mutation. Lastly, nonsense mutation occurs when instead of an amino acid, a nucleotide is coded with the stop codon. So when one amino acid sequence such as this has one nucleotide base coded with a stop codon, the amino acid sequence will now look like this. While mutations occur in the micro level, it produces huge differences in the traits and the characteristics that an individual can express. Common includes natural plant color mutations, such as this, medical conditions in animals such as polydactyly, syndactyly, ocular albinism, and hair color patterns can also occur. Similarly, other forms of animals may also experience hairlessness, albinism, and kinds of anatomical variations. Learn more about mutations as you explore the following references. Thank you for watching.